Vos avisame, ¿no? No, pero me tengo que parar acá, ¿no? Acá hay que pararse. Vamos, vamos, que la última vez, Pablo. Bueno. Bueno, no.
¿Qué? ¿2 minutos?
No, no.
No, bueno,
El
sí.
micrófono está bien.
Uy, que luego.
Viste que no son divinos. Aparte no está como queda el fondo negro.
Bueno, vamos a empezar.
Sí, ella me avisa porque ella streamea. ¿Ya estamos en vivo? Buenas tardes. Buenas tardes, mi nombre es Paula Pulso, soy investigadora de la Facultad de Medicina de la Universidad de la República acá en Uruguay. Y les doy la bienvenida a la conferencia de cierre del doctor Gabriel Mindlin de la Universidad. Buenas tardes en Argentina. Buenas tardes, mi nombre es Paula Pulso, soy investigadora de la Facultad de Medicina, Beyond the Lab, Naturalistic Approaches de la Universidad de la República acá en Uruguay. Esto, y les doy la bienvenida a la conferencia de cierre del doctor Gabriel Mindlin de la Universidad. Buenas tardes en Argentina. Otros países del mundo. Buenas tardes. Está organizada por estas personas y tiene el apoyo de un grupo que lo vemos aquí en este momento y cuenta con el financiamiento de la Universidad de la República, de Company of Biologists y de PDCIVA. Bien, now I'm going to switch to English. I'm going to introduce Noel Federman. She is from IBIOVA Max Planck Institute in Argentina, and she's going to introduce Dr. Gabriel Mindlin.
Thank you. Thank you, Paula. Okay, it's a great pleasure for me to introduce to you Gabriel Mindlin. Gabriel studied physics in the University of La Plata, and then he moved to the US during his PhD in the University of Drexel in Philadelphia. He was a professor in the University of Navarra and also in the University of California. And now he is a full professor in the University of Buenos Aires and also a researcher at CONICET. His research produce dynamical models that allows to reproduce the complex songs of birds. This dynamical model were applied not only at the level of the muscles, but also at the level of the neurons, and allowing to Gabriel to talk with the birds and sing with the birds. He wrote a lot of books and also more than 100 of research articles. And in 2010, he was selected as a fellow of the American Association of the Advancement of Science. So it's a great honor for us to have you, Gabriel. Thank you.
Should I use this one? Okay, well, thank you very much, Noe and Paula, for inviting me here. It's a great privilege. I remember the schools I attended when I was your age, and they are responsible for many of my scientific friendships all across my career. So I hope you are creating these bonds that are typical of these schools. And before I start this presentation, which is the last of this school, I would like to join me thanking the organizers for this wonderful school. Because it's a lot of work and it's highly appreciated. And when you are my age, you'll know how much work it takes. Okay, so I also want to thank for the openness of inviting, for closing this school to a physicist, not a biologist, and also a person who in principle doesn't work that much outside the lab. But I want to tell you some stories which could be of some interest given the topics here. The reason I'm telling you this is because I come from not only the area of physics, but the area of applied math that is behind physics, which is nonlinear dynamics. So the kind of questions I was interested when I started working in birdsong, which is more than 30 years ago, is how the brain and the periphery interact in order to generate a complex behavior, and how much of the complexity arises from the instructions that are given by the brain, and how much from the nonlinearities of the peripheral device that is responsible for the behavior. So my work for many years was theoretical. And in this dialogue between physics and biology, which is trivial, how the brain and the periphery interact in order to generate a complex behavior. Simplify, and you know much of the complexity arises from the instructions that are given by the brain, and how much from the nonlinearities of frame things peripheral device that is responsible for the historical discipline where you study this. So my work for many years was theoretical, one cell to what we are now, and all this complexity cannot be just simplified. So in order to do reductionist approaches and models in biology is very delicate. So one of the issues I was very interested in all along these 30 years is how you test the pertinence of low-dimensional approximations to biological problems. And some of the stories I'm going to be telling you today have to do with that. So most of my work was mathematical in nature, and then how to create the technology for measuring the parameters that are important for those models. So it was heavily about building labs and developing the surgical techniques and the electronics needed to measure these subtle things like the activity of tiny muscles in the vocal organ of birds and stuff. So it was not that I was working outside the lab for many, many years. But of course, life is full of surprises. And what happened to all of us a few years ago was COVID. And COVID was responsible for, in my case, going outside the lab, because in Argentina, we had a very long quarantine, almost 2 years, 1.5 years, without people being able to attend the lab. And during that time, there were students that had to write projects, theses, special laboratories, and we don't want them to delay 1.5 years or 2 years their careers. So we had to invent something to keep people busy and developing projects and doing stuff like that. And so it occurred to me, since I live outside the city of Buenos Aires, I live far away, like, well, many kilometers away from the city of Buenos Aires where I have my lab. I live very close to a state park, which is called Parque Pereira, which is a biodiversity reserve by the UN. So I thought, well, maybe we cannot meet in the lab, but maybe we can meet in the field. So let's see if we can do something that is pertinent to the projects that these people have and do some work in the field. So what I wanted to do was to generate projects that were very simple. And in principle, if I want things to be simple, I needed to work with species, which are very easy to find. So I tried to focus on the most ubiquitous species that you can find where I live, which is the Chingolosonotrichia capensis, which is this bird that we have in here. And I'm going to be talking about this tiny guy. Okay. Okay. So this is this bird I'm going to be talking about. Sorry for the movement at the beginning. Let's listen to it for a while. Okay, and this bird is a learning bird. I'm going to be talking about learning birds, sign birds, but it learns only one song. And what I'm showing in these graphics here is what is called a sonogram. It's a pictorial representation of the song. So it's like the script. You have in the vertical axis the frequencies, in the horizontal axis you have the time. Each spot there is where you have sound, and you have very simple lines where you have very simple, to it for a while, almost tonal. What is called a sonogram. It's a pictorial representation. So this is the guy which is more easy to find in the place where I live. As I was telling you before, just a comment, although if you're a biologist, this is trivial to you, but in principle, when I talk to physicists, I have to explain. Why bird song? Well, because songbirds are an animal model that is ideal to study learning of vocal communication, okay? So vocal communication is something that in humans we have to learn. We listen to a tutor, mom and dad, and we learn the vocalizations, and that is not shared by many species. But Osine birds, which account for 40% of the bird species, need to be exposed to a tutor in order to have the proper vocalizations of the species. So there are like 10,000 species. 4,000 of them are Osine birds like canaries, zebra finches, or cingolo. And of course, they all have very different brains. Some of them learn one song, like the cingolo, maybe 2 or 3 in some cases. Some birds have humongous repertoires, but in principle, they all share some common architecture in their brains, which allow them to reconfigure the connections during the process of the learning. So the questions typically are which are the dynamics of learning, and this one, to me, is very interesting, is how much of the complexity in behavior is conditioned by the biomechanics of the production. Of course, when you say learning, and you talk to people that are neuroscientists accustomed to work in the lab, they will say, well, if you keep a bird in isolation, they won't sing the typical sounds of the species. And of course, that doesn't happen ever in nature. So what does it learning mean if you go to the field? And it's interesting because I never paid attention to that until the pandemics. So let me show you one of these tiny projects that we did with these physics students. We put 4 microphones connected to one Zoom recorder, and we measure the sounds with these 4 different channels. And of course, the time it takes the signal to arrive to these different microphones is slightly different, and you reconstruct the position of the sound source just by accounting for the time delays. So here you have in these different dots, so these are the microphones, and in these different dots, what you have are the positions of the birds that were singing, and here you have an adult that is keeping the territory, okay, this is during the spring, so the bird is singing over and over, maybe 4 times a minute, is singing this song. The song is characterized by these introductory notes that are typical of this bird and a rapid trill. And then you have all these juveniles that will keep, their positions there, and they will start to repeat from time to time, okay, their own vocalizations, which in this case are somehow similar to these vocalizations that you have in here. So they're not identical. This one has only 2. This one has 3 in some range of frequencies. This has 3, but in a wider range of frequencies, and so on. And they will start to sing, and for 3 or 4 months, they will sing close to one particular tutor. They will stop vocalizing at the end of January. And then in the next year, you will see that what was just a draft of a sound, of a song, okay, during the spring, then by the next year was a perfect, very confident set of syllables with a complete trill, okay? So this is what learning is all about, no? Identifying a tutor, patiently repeating, and then eventually the next year having some crystallized song. But of course, walking every day for hours in that place because I couldn't go to work, I started to recognize these particular individuals that live in Parque Pereira Iraola. So these are highly territorial birds, and what I'm showing you here are the sonograms of so different individuals. I was scared, man. You heard it too, right? It's not. Good. Good. So these are different individuals, and these different individuals, you can recognize them because the song of these birds, as I was telling you, have a particular theme and then a trill. The trill is characteristic, so this is what we call a dialect. They're characteristic of a wide region, okay? Several hundred kilometers with more or less similar vegetation and so on. But then the theme is characteristic of each individual. So this guy will repeat over and over this pattern, this one here. And of course, they are very highly localized. So after a while, you recognize them. So I thought, well, what can we do with this? So I started to look in the literature what was known about the species, and it's not so much because there are 10,000 species, and one little bird that is not so special in South America is not so widely studied. But fortunately, there was this guy who is very famous, Fernando Notebaum, who was an Argentinian who also got stuck in Argentina in the 60s. And it's interesting because at that time there was the military service. You were drafted. I was drafted, okay, when I was 18 years old. Everyone was drafted to go to the army and service 1 year in the army. It's what we call in Argentina, la colimba, okay? And if you wanted to escape from that, you had to ask for special permissions and so on. Fernando Notebaum, he immigrated to the United States very young, but at some point he was called, come on, you know, make it clear why you never served the army. So he had to come to Argentina for several months to explain his situation. And he was bored like hell. So he would walk by Parque Pereira. And since he didn't have any special equipment, he would take handwritten notes of what the birds sounded like. Now, keep this in mind. We have a computer in our phones. We can, you know, record anything. We can use Burnett, Merlin, whatever. We can do amazing things with the phone. At that time, in the 60s, we didn't have digital recordings. Digital recordings are from the 70s, okay? So you could record something in a while, but you have to go with these huge tape recorders, which were very heavy, very expensive. And of course, you couldn't process them online. You couldn't compute a spectrogram like this because you didn't have digitized data, okay? If you wanted to have something similar to this, you had to use a special equipment that was developed in the Second War, what was an electronic device where you would record the sound, you would play it over and over, you would filter it, and you would burn with a hot tip a thermal paper, okay, and you will build a spectrogram there. Of course, Notebaum didn't have this in Parque Pereira. So what he did, check this out because this is amazing. He would take these handwritten notes, and then he returned to Rockefeller in New York, and he calibrated himself. So he played himself a song. He will build a spectrogram there. Handwritten notes. Of course, Notebaum didn't have this in Parque Pereira. And compare the two drawings and send it to the editor to show that he was very good taking notes. Try to do that now. Amazing. So we had these very trusted, spectrograms, and my question was, what are the same themes that are sung today? So it was a very easy project for me. So how can you do that? How can you test whether these were the same songs that we can hear in the field today? So what we did is to develop some tools with artificial intelligence to compare patterns. So what I'm showing you here is the architecture of two neural networks. So this is the typical convolutional neural network that you use to classify whatever. If you put two together, it is what you call a Siamese network. And you train this not to classify anything. You use it to define a matrix. How you do that? You have all these units, the typical neural network, and at the end you have a layer which has no meaning whatsoever. These are the coordinates of an abstract space. But you train the network so that if you have two patterns that you decide that they are different, you map them far apart in this last representation. And if you have two patterns that you decide that are similar and belong to the same class, you train the network to map them close together. Therefore, in this way, you define a metric that you can use to compare patterns. So my idea was to compare the patterns of the songs that were sung in the 60s with the patterns that are sung today. Only problem, if you train a neural network to identify differences between this and this. This is what I compute now with a digital device, okay, with computing a spectrogram with, I don't know, with Audacity or any software that you want to use. And these are the handwritten notes of Nottebohm. A neural network will say, well, this is one kind and this is the other kind. It won't pay attention to what the frequencies are doing. It will pay attention to that one is written and the other one is computed, if you want, right? So you don't have control over that. So what I had to do is resynthesize this and process them with the same tools that I applied to the songs today. So we generated with models that I'm going to describe in a minute or in a few minutes, the themes that Nottebohm found. No, the themes that, so here is the synthesis of the themes that are sung today, and this is the synthesis of the songs according to the handwritten notes of Nottebohm. And when you do that and you process them with the neural network, you found that, of course, these different birds are mapped into different sections. This is a two-dimensional representation of this last layer of neurons. But if you pay attention to that, you find these beautiful intersections of some themes. And this is what I call the hits of the 60s. Okay. They were very popular in the 60s, and they are still being sung today. Fortunately, Nottebohm also kept record of how many birds were singing each of these themes, so I know that these were also the most popular themes there. So they survived probably because they were sung by a lot of individuals, and some songs that were sung by just a few individuals with this decaying amount of birds in this region were lost. So there are some songs that are preserved and some themes that are lost. So there was another project for the students to keep themselves busy during the pandemics. And then I thought, well, there is something also interesting that is the following. What about the themes that were lost? Can we reintroduce them? Now, I had no idea at that point whether these birds, a juvenile, could learn from some synthetic tutor. And to me, that was a very interesting question, because to me, whether a song is realistic enough to cheat a bird or not is a central question of my area of research, and I'm going to be describing that in a minute. So what about the possibility of introducing this? How can we do that? How are we doing so far? Perfect. Okay, so let me show you how we can do this synthesis and how we can try to teach a juvenile to sing like we want. Here is the physics of birdsong in 15 seconds. Never repeat that you can learn this in 15 seconds, because it took me like 30 years of grant money and students and stuff to do it. But basically what happens is that when the bird is breathing, air is coming in and out, but then at some point, the bird is going to push this labia together, and you are going to elicit oscillations in this labia, just like what would happen between two pieces of paper if you blow in between them, right? Basically, when you modulate the airflow, you generate a derivative of the rate of mass injection of air in here, and this is the origin of sound. Of course, you can, while you are doing that, while the bird is doing that, is pushing this air, that is a special gesture. I'm going to expel a lot of air. And also you can move some muscles in here that will change the configuration of the settings, stretching the labia or releasing them, and in that way, controlling the frequency of the vocalization, okay? But the physics is not complicated. I'm going to just write equations to intimidate you, right? To show that we physicists can do wonderful things. But actually, it's just for laziness, because it's the kind of a transparency that I use for other audiences. But basically, what you have is that you have to model the physics of the motion of the labia in here. And although these look strange, these are Newton's equations for labia. It's nothing that goes beyond physics 1. Here, also sounds very intimidating, but it's even simpler. It's just what happens when you filter a signal, because this sound wave come in this tract and then resonate with this cavity there, and some frequencies are enhanced and some frequencies are dissipated. And this is the physics of the filtering in here, okay? And of course, what you need to find is the behavior, the time-dependent behavior of the parameters of this model. For example, this restitution constant, which is telling you which frequency the oscillations are going to take place, depends on how stretched these labia are. And you control that by contracting some muscles. So if you want to increase the frequency, while the bird is phonating, you have to contract this muscle there that will stretch the labia, okay? So then you will have that the frequency of the vocalization goes up. So you have to impose some time dependence in the parameters, like the pressure and so on, and you synthesize some sound in here, which in principle sounds similar to a song. How similar? This was a question that took us a lot of work with Anna when Anna was working at the University of Chicago in that Margoliouth. I'm going to tell you this story, which is very dramatic to me, only because it has a happy ending. But just to show you, young people, how dramatic a scientific career can go, let me tell you this. I had been working in the physics of birdsong for many years by the time we did this experiment, and I had published in many highly respected journals. And everyone in the field of birdsong knew that I was the guy doing the physics of birdsong. And I was going around saying the typical physicist story: very low dimensional model is good enough, right? So we go to the lab of Dan Margoliouth, and I say, it is so good that if you test with a bird, okay, and you ask a bird if this is a real song or not, it won't find a difference, okay? So how you do that? Well, songbirds have these highly selective neurons in a region of the telencephalon called HVC. That means there are neurons there that are used for the production of the sound, that if the bird is asleep and you play the song back, they will spike, okay? So the experiment was very easy. The bird is sleeping. You do a playback with the song of the bird, and it will spike. And if instead of playing it with the song, you play with the synthetic song, it will spike. Very simple experiment. And I was so sure of it. So I go to Chicago, and I tell Dan Margoliouth, low-dimensional models and Wi-Fi physicists are so cool. This is just two equations and so on, and it will work. And he was saying, like, no. I said, yes. And he wasn't even going to allow Anna to do the experiment, and we insisted with the experiment. And in the experiment, you do this. You play several times the song, and if you play the birdsong song, which is this, okay, you will have some spiking. If you play the reversed song, okay, you have no response whatsoever. If you play this conspecific song, there is no response. So I did this trick of generating a synthetic song. We played it, and there was no response. Nothing. Zero spike. I didn't know what to do. No? So I just wanted to hide, okay, because it was not just an experiment going wrong. It was the hypothesis of a whole line of research going wrong. It cannot get worse than that. It cannot. Of course, I'm telling you this story because then we tried again. And for the questions, if you're interested, I can tell you why. When we repeated the experiment slightly modified, we found that, yes, the synthetic song was capable of eliciting responses like the birdsong song. So here you have the okay, synthetic and real song, and you have responses. It's very interesting what we had to change here and what is the meaning biologically of what we had to change. This is another lesson that I learned it here in the hard way. The reason I failed in that case was for my prejudices. Okay, since I found the nonlinear part of the problem so interesting, I focused on that. And since I thought that the linear part was so stupid, I didn't pay so much attention to that. Well, but birds do pay attention to that, but because that's the way in which they convey information, for example, about their sizes, right? Not a small thing. But anyways, so if you revisit your prejudices and you correct them, and you learn some humility, then nature compensates. So this was in the lab, but now what I wanted to do is whether to test if birds in the wild could also be cheated by this kind of synthesis. So what we did is build with some Arduino technology, programmable processors, some recorders, devices that would do playback, and started to think about asking the birds in the wild whether they thought that our syntheses were good or not. So in here, what I'm showing you is a theme of a bird that is defending a territory, and then I would go to the field and I will try to invade the territory either with a real song or with a synthetic song. So here is, well, what happened? That sounds better than mine. Well, okay, there's something, yeah. These two will be very similar. Okay, and what we did is we generated a whole bunch of songs that would correspond to good copies of the songs that were recorded in the territory. Okay, good copies of the songs in the territory. And we tried to see what was the result of playing these songs, good copies, as invaders that were to a guy that is defending a territory. So what happens when a bird is defending a territory? Well, if the bird is singing 4 songs per minute, if you start to play for a couple of minutes the song of an invader, it will start to sing many more songs per minute, 6 or 7 songs per minute. It will try to sing more, okay? And then when you stop with the stimulus, it will take it like 10 minutes to go back to a baseline, okay? So here what I'm showing you is what happens when you play the real song and what happens when you play the synthetic song, compared to what happens if you play the song of a specific bird. Okay, here if you have a test, you have that you go below significance, so you have a below significance in terms of differences, okay, and then you recover the normal song. So the responses of the birds to synthetic songs or real songs were equivalent. So what I thought is, well, what about the juveniles? Do they need to interact with a real adult bird, or could they be cheated by one of these synthetic robots and try to learn from them? So what happens when you play the song of a synthetic robot to a juvenile is very interesting. Let me show you this movie. Sorry for the quality, this is just a phone, okay? We're in pandemics and it's just a phone. So here's what happens with a juvenile, okay? Only 2 very shy upsweeps, okay? You see how the stimulus encourages the bird to try to do better, a more persistent and confident song. And this is what happens with the juveniles. They will find something interesting and they will go there and try to convince the box that they are doing quite good. So what we did is started to go in the park and see where the juveniles would get together, because chingolos are very interesting. They are with mom and dad in the nest, then they fly away, when they are kicked away actually, and all the juveniles go together to some region of the park and they start to play around, right? until they identify a tutor and start to sing with them. So we found where the juveniles were meeting in the park, and we built these 3 robots that made this specific song that was extinct fashionable. So they were very enthusiastically singing these robots, okay, these songs during 3 months. Juveniles were crazy about it. But we have no idea whether they take it as a model. So what we did is place these boxes, okay, play these boxes there, and they were playing and recording at the same time this song that was a song that was lost. What did it have different from other songs? That this second syllable was a downsweep. Every other bird that is recorded in the last 5 or 6 years in the park has the second syllable like an upsweep. But according to Nottebohm, it was possible for these birds to do this different syllable. We made it fashionable through these robots. We had no idea what was happening because we didn't spend much time there. But at the end of the summer, there it was, a juvenile singing to the box with the second syllable with the downsweep. So we're super happy about it because it told us that this signature that was not sung in the park was finally captured by a juvenile here. The only question that I had was whether that was an anecdote or not. Let me first tell you the quantification that it was the syllable. Here what you have is the projection of our neural network that was trained with regular songs when processing the information of these juveniles. And here you have what happened with the very young guy. The same guy, a little bit later, when it started to make the first downsweeps. But this is what I found very wonderful, that in the next season, what happened with the very young guy, in the same region, the same guy doing song, a little bit later, the second syllable like when it started to make. So in principle, either there were more than one juvenile, there was more than one juvenile who had learned it, or maybe this juvenile was teaching new birds. We didn't know because we didn't ring them. But at this point, I was obsessed with finding the guy, right? I wanted to find where the guy was. Okay. And I'm very proud of this because here it is. Pa, 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 pa. Here it is. Now it's an adult singing very confidently, okay? The song that we taught it. And I was very happy because the story got to National Geographic. It was a story covered by them. Yeah, and actually, this is the title of the talk, but they chose it. The robot taught these birds a long-lost bird song, which I found it kind of cool. I think that beyond the anecdote, the idea of using these kind of synthetic songs to generate specific stimuli has a lot of potential. Here, what I'm describing is a work that we're going to doing together with Cecilia Di Marsico and Juan Carlos Reboreda and Camila Stasta, who is the grad student doing the work. And it's a wonderful story because, you know, Juan Carlos Reboreda studies parasitism in birds. The story is like this. This is the tordo músico, one of my favorite birds in La Pampa, by the way. I love this bird. I'm sure that in Uruguay you have it too, right? The tordo músico is a guy that likes to fly around with all the friends and they're singing together. It sounds like they are coming from a party, okay, and they don't want to go home. It's a very messy bird, but it suffers the parasitism from this guy here, which is the screaming tordo, no? The tordo. The screaming one, this is Rufo axillaris. We call it the Rufo. The interesting thing is not that the female puts the eggs in the nest of the tordo músico, which is a more or less common story. But it is a species that is specifically a parasite of this species. And the juveniles look identical, but the sound of the juveniles, okay, when they hatch and so on, is different. Interesting enough, they hacked the system of the female musico, so the female prefers to feed the parasite and know its own babies. It's kind of sad, right? So what I'm interested is in finding which are the acoustic features that were used to hack the system of the female musico. And what we're doing is using physical models to generate first a good copy of this synthetic song, that they're equivalent. We showed it last season, and now what we're trying to do is to change the parameters to go from these kind of vocalizations to these kind of vocalizations to find which are the specific features that they have to do. But one interesting thing is that the model is more complicated. You have to have a higher degrees of freedom. You have to have like chaotic motion in the labia to be annoying enough, okay, to urge the female to feed you, right? So it's interesting. It's something about being really annoying, which is the key to this. We say in Río de la Plata, el que no llora no mama, okay? Another application of these techniques in the field is what we're doing together with Kini Rosler and the people of the Programa Patagonia. They work in Santa Cruz in a program that used to be run by Aves Argentinas until last year. Now it's a foundation specifically the Macato Viano. Macato Viano is this very charismatic little duck here. It's a macaque, it's not a duck, but I call it a little duck. That was discovered in 1974, okay? But it's highly threatened, okay? Particularly, there were a few thousand species until one Huron came to the region and got rid of 20% of the population in one night or something like that. It's an incredible story. It's adapted to a very specific region, and there are just a few left. And what we want to do is to use these artificial intelligence techniques to follow the song of every particular individual. Okay, we want to track every particular individual, and we're also using it for another threatened species, which is Rallus antarctica, this gallineta, which is also a very threatened species of birds in Santa Cruz. So in summary, and to conclude, what was interesting to me is whether minimal models in biology are pertinent or not. And this is an interesting thing. Probably you are modeling your phenomenon, or you are going to be interacting with the modeler. And it's very interesting to know how to interact with someone who has a quantitative approach, because you have to understand that it's not a question about language. It's not a question about, well, a physicist knows mathematics and biology knows less mathematics, or the biologist knows a lot of biology and the physicist knows less. This happens, but it's not the main problem. The problem is that the physicist thinks that if you're not finding the actual minimal mechanism, it's not being a good physicist. And the biologist says, if you're oversimplifying, not paying attention to the real complexity of the system, maybe you're getting rid of 100 million years of evolution, okay? So what everyone thinks is rigorous is different. So you have to keep in mind that it's not a question about ignorance, but it's a question about what is interesting and deep for the other person. And if you understand what the other person's mindset is, you will be able to interact more, okay? Because it's very easy to find a different perspective as synonymous of ignorance, and it is not. It's just a difference of culture, okay? So to me, in order to find whether a minimal model in biology was pertinent or not, had to be of asking the birds. And the best question is in the field. Can you cheat someone in the field? Well, in that case, that's good enough, right? Okay. So can a juvenile take a synthetic song as a tutor? Yes, and that thing is very interesting. The other thing that got me thinking is that you usually think about biodiversity like a genetic question. But animals have cultures. This case is very obvious. It's a behavior that is taught from one generation to the other. This is culture. So when you are talking about biodiversity, you are not talking only about genes. You are also talking about culture. So I think it's interesting to take any kind of action, citizen science, whatever, that keeps this culture going, because we're jeopardizing it all the time. You heard that too, right? and I think that we have to preserve it somehow, okay? There is culture in the animal kingdom, and it's our responsibility until we stop destroying it, at least keeping it, okay? So artificial intelligence and dynamics together for conservation, these applications that I was telling you about, and the future work is going beyond species identification to individual identification. I think it's going to be making a big difference in terms of conservation projects. And with that, thank you for your attention.
Thank you so much for this talk. Any question?
So very stuck, very nice work over the pandemics. It was a hard time. I'm working with crabs, with Tom Sick. Do you know Tom Sick? And we discuss a lot about the why of the model, not the how, not the like the reach, the why of the model, because he's not very convinced the models do anything like in particular. And I, as engineer, I very like models for explaining things. So do you have some?
Yes. I do have a question for that. I think that the advantage of building models is that you're identifying which, and actually I think this is such a deep question at this moment, where you have a paradigm that is completely different for dealing with problems, which is artificial intelligence, that is interesting to have a clear position about that. A model assumes that you identify which are the important variables and that you understand the mechanisms that relates them, okay? So when you have that, you can test hypotheses. You can change parameters that you can interpret, and you see whether there is a hierarchy of importance in them in order to obtain a response, okay? In my case, for example, in order to elicit a response of the bird that is asleep and recognize that as its own song, it was very important the size of the animal being captured. If you change that parameter, then the response decays a lot. But if you change another parameter, maybe the response is indifferent. So which are the important variables? Is there a hierarchy of importance of specific questions? So when you build a model, what you're doing is you are making clear your hypothesis, and you make the path of how to test them systematically. So the importance of the models is not to test that you are smart enough to reproduce a result, because that for nature is anecdotal. I mean, I am a good modeler, you are a bad modeler, or vice versa. Okay, that doesn't interest to anyone. But if you have a good model, you have a way to ask good questions in terms of mechanisms. And with a model is the best way, because a model synthesizes a mechanism. Now, artificial intelligence, can they make a better synthesis than my models? Of course they can. I can make a synthetic sound much more similar to the real one, and I can put two adversarial networks compete themselves and generate a synthetic sound that is completely impossible to distinguish. So what? What do I learn from that? Nothing. I make a model like this that is good enough, and I say, well, this parameter represents the size. Is the size important for the female? Is this sexy for the female or not? And you identify the particular feature that is important for the animal. Without a model, you cannot do it. You just change phenomenology. With a model, you change mechanisms. This is the way I see it.
Okay.
Fantastic talk, first of all. You dedicate a few minutes to explain why study the bird song? I think that we are tired to explain what we do, and we sometimes don't want to do it, but it's important to find the society, to have them in our side and make more relevant what we do. With this in mind, and this juveniles try to do the song, we can learn with this model about how we learn to talk.
Yes. So the answer is yes. There are many similitudes in the process of learning. They were pointed out by two wonderful women. One was Patricia Kuhl, and the other one was Alison Dope, who passed away maybe 5 years ago. She was a psychiatrist at UCSF, and they wrote a very influential paper called, well, I don't remember the exact title now, but it was a comparison between juvenile humans and birds, and the critical periods, the building of a template, the successive practice, what happens when you close this critical period in terms of your ability to learn, which maps exactly. I don't know if, for example, if you are a Spanish-speaking person, you notice that for Chinese people it's difficult to pronounce the R, and they will misplace it with an L. And it's not just a question of producing it, they just don't perceive it, because if you are not exposed in a particular period to the difference, then you cannot even perceive it, much less then try to reproduce it. The same happens to us when we try to say the difference between sheet and sheet, okay, which is so hilarious for an English speaker, and I cannot do this, but I cannot even perceive that I'm not doing it right? Because I wasn't exposed to those vowels at the critical period. So there are many parallelisms that you learn through that. And just the question of the process of the dynamics of learning is very illuminating in terms of path. Of course, it's basic science, and at some point society has a right to say whether they're interested or not in using that as an animal model. For example, NIH at some point stopped to use it as an animal model for deafness diseases, and you have to live with that. But I agree that it's an important point to if there are connections of translation, to stress them. Yeah.
Okay, thank you for your talk. It was pretty interesting. I was thinking about you said you can like make something with AI o sea, in the way the birds cannot separate them from a real individual. Can we make the opposite, or is it possible to create or train a model to the point we can't distinguish it from some animal model? I mean, in the way they behave or respond to certain stimuli, and then knowing how we build that model, like know what are the circuits or eso, ¿no? Por ahí, tipo, sacar qué es lo que pasa, what is happening in the brain by like this interpolation or this, eso. Bringing the AI models into the biological terrain and not vice versa, no?
It sounds like a very exciting idea. So I never thought of it systematically, but sounds very appealing to me and makes a lot of sense. I haven't seen anyone exploring that. Profit from it. It sounds very interesting, and yeah, makes sense, makes a lot of sense. Of course, the more disruptive these ideas are, the more fiercely they are going to be fought and resisted, okay? So when you are deciding what to use as I'm talking for personal experience, the more disruptive you are, the higher the price is if you get it right, and the tougher the resistance is at the beginning. So I would suggest to use a very disruptive idea like this, like to keep it for when you are a little bit more established, and not to use it in the critical periods of your career when you have to jump from one position to the other, and a critical mistake is highly paid. But this is the typical idea I would invest many years on the moment I have a positioner. Yeah. I'm very opinionated. Don't make any, don't take me seriously, right? But
So great talk. Like I use models and like sometimes my models mismatches like what I want to see. So like, have you ever seen your models a mismatch of like another bird learn like another like a song from the other one?
so if you're, maybe I can rephrase your question whether I have proposed a model that failed miserably. Yes, all the time, all the time. I would say that actually none of these models was the result of a lucky inspiration. They are the result of tuning a series of failures, of dramatic failures, until you find what is good. And the process of learning from those mistakes is the best that can happen to you. And it's related to the question of your friend. The good thing about the model is that you are testing a specific hypothesis. So when your model fails, it's cool because you are learning something that you are thinking badly. Okay, so it gives you the opportunity of tune your thinking about it. So yeah, they fail all the time, and before these models, for many species, I tried things that didn't work. I was telling you about songbirds, but there are 10,000 species, and I'm very interested in the comparison between learning birds and non-learning birds. And the non-learning birds have different architectures in their seedings, and they have different physics, and we've tried for different species, different models, and sometimes they just don't work. And when they don't work, maybe you're missing something, you go to the anatomy, and you find, of course, it didn't work because my hypotheses were wrong. So, yeah.
Any other question? Yes.
I have another question. Do you think that this reintroduction of different old songs could have a biological impact in the ambient?
It is a very good question. I've been asked that, and it's interesting. In my opinion, no, because it was a song that we know that in that place it already existed. I'm still monitoring it, and what, for example, happened to me is that we found for 2 years these birds singing this song, but for example, this year we found no one singing that song. So probably it decayed. Of course, I would be scared to death if all of them started to sing this song, and I probably wouldn't be giving the talk because I would be hiding. That didn't happen, but my rationale in that is you're making a very small intervention, which is just this particular acoustic feature of something that you knew that existed. So my estimation is that not, but it's a pertinent question from the ethical point of view. Yeah, I take it as a valid observation.
Any other question? No? Okay. Thank you so much, Gabriel, for the talk. Okay. Okay, we have one announcement. We are going to take a picture of all of us